Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Hi all, welcome to another episode of Beyond Lab Walls. I'm your host Nicole, and I'm excited to get into this very special episode. Before we introduce our guest, I want to welcome today's co-host, our high school summer intern, Amina Aslamir. Hi, Amina.
Hi.
She has spent the summer working with the Salk communications team through the Heidoff Brody High School Summer Scholars Program. This has been a really important program run through Salk's education outreach team. They invite local high schoolers to train at Salk and explore careers in the life sciences. So traditionally, those students worked in labs on campus, but this year the program was expanded to include internships with the communications team and our Office of Technology Development. So I think this has just been a really great way to show students that you can work in science and contribute to this field without actually being a researcher in a lab. So, there are just a ton of other careers in science, and it's been really fun to share the world of science communications with Amina this summer. So we're glad to have you here today. And speaking of unique careers in science, our guest today has one of those. She's the program manager of Salk's Harnessing Plants Initiative, which is a really unique program here at Salk. Of course, the Institute is very well known for its work in health sciences, but our plant biology research has really been blooming, pun intended, in recent years. So yeah, the Harnessing Plants Initiative is really capturing the power of plants to help fight climate change. So we'll talk all about how their science is being used to save our coastlines and support our food supply. And we'll be guided through it all by our great guest, Kay Watt. So without further ado, welcome, Kay. We're so excited to chat with you today.
Yeah, I'm so excited to be here. Thank you both for having me. I'm looking forward to a very invigorating conversation about the challenges of climate change and also what we can do to mitigate climate change moving forward.
Yeah, it's such important work. So Amina, why don't you get us started?
Hi. Before we learn more about your science, we'd love to get to know more about you. Where did you grow up and what was your life like there?
That's a great question. So I'm not from California originally, although I have been here for over a decade. So I feel like I kind of counted as an honorary Californian by this point. Whenever I go home to Massachusetts, I only go in the summer because I'm not ready to handle Massachusetts winter again in my lifetime yet.
Yeah, I'm the same.
But I grew up originally in Massachusetts, so plenty of cold, snowy winters. I was born and spent my childhood south of Boston on the South Shore, where I lived in the suburbs, but I lived right next to a nature preserve. So we would do a lot of nature walks, and we also had our own garden growing up as well. We grew corn, we had beans and peas, just whatever we wanted to plant. And of course, pumpkins for Halloween.
Yeah. Oh, wow. Lovely.
Yeah, it was lovely. So I mentioned my family did a lot of nature walks, especially in the winter. We would go on walks in the woods where all of the leaves have fallen off the trees. Maybe there's a couple of pine trees that are still green, but there's snow on the ground. So you have kind of a gray sky, like the black bark of these trees, and then a couple green pine trees. And then every so often you would see a couple of really beautiful bright red pops of color that were flittering through the canopy, which were cardinals, which was one of my favorite birds growing up.
Yeah.
But one of the things that I realized, I think in the last couple of years, is that cardinals aren't native to Massachusetts. So in the 1900s, they were more native to Kentucky, and they were called a Kentucky cardinal. But since then, their range has moved further north. So when I was growing up in the 1990s, it was in Massachusetts, and now by the 2020s, their range has extended all the way north into Canada. And the reason that they're moving and changing where they can live is in due part to climate change. So as winters get milder, they can spend their time over winter in these new places.
That's wild because I also have that memory up in Jersey, those cold winters, and the cardinals are so beautiful. But yeah, it's strange to think then, looking back, that this like beautiful part of our childhood was actually sort of a warning of climate change, even that long ago now.
Yeah, I don't know what the opposite of rose tinted glasses is, but I think now we're both wearing them a little bit. Yeah.
Oh man, wow.
How did you first get interested in the sciences?
Yeah, that's a good question. So I was really interested in nature and agriculture, but I wasn't interested in science so much until after my bachelor's degree, when I served in the Peace Corps. And the Peace Corps is a very small group of volunteers, of United States citizens that work with the federal government to do outreach in communities in different countries across the world. I spent my time in the Peace Corps in Panama, where I lived in a very rural community working on sustainable agriculture. And at the time, as like a 22-year-old who was fresh out of college, I had the maximum amount of idealism I think I will ever have in my life, and was very sure and confident that I could do anything I set my mind to. So I was very humbled by my experience there, where we were working with almost no funding, very little supplies.
What did that look like day to day?
So I lived with a host family for the first 3 months that I was there, and that started about 6 a.m. You'd hear the chickens outside or the pigs that were like roaming by. We'd get up, go outside because the roof was made out of zinc and it would heat up very quickly in the sun, and the house was made out of cinder blocks, so there was no like cooling down inside the house, so you spent most of your time outside until sunset. So a lot of the time was just spent sitting with people, chatting with them, getting to know them, getting to understand them, and building relationships, which from my perspective as somebody who is like very motivated to like sit down and get the work done, seemed like not a waste of time, but seemed very slow in comparison to what I thought I would be doing. But it ended up being the most important part, is building those relationships, hearing perspectives, understanding what people think they need, because usually what they think they need, they're the experts in their own lives. So they're the ones who do know and can tell you, rather than you coming in with an idea and kind of saying, this is what you need. So one of the main problems that I was brought in to solve was helping to increase the health of coffee trees in the town itself, because coffee was kind of the exported good. But I worked with farmers specifically in sustainable agriculture, mostly working on coffee farms to prune trees in a certain way so that pathogens or different bacteria that were present naturally would have less of an opportunity to infect and take hold in these trees. But at the same time, I did some work with the school and with people to be able to set up their own gardens, because where we lived, people would grow subsistence food. So that meant they would have their own rice paddies, they would grow plantain, they would grow banana. So that's kind of what people relied on to be able to sustain themselves and their children. But unfortunately, even though it's a tropical country and you don't have harsh winters, a lot of food varieties or crop varieties couldn't actually thrive there. So if people wanted to eat like a tomato or a cucumber, you could plant them and hope that it did well. And 9 times out of 10, it would not do well. So it would succumb to a disease that it was facing, or there would be like an extreme rain event, or there just wasn't enough nutrients in the soil itself, because it's a really thick red clay rainforest soil, which is very nutrient poor. So if people did want to eat something like a tomato or a cucumber, they would have to send somebody in their family 3 hours on the bus to a town to be able to pick them up and then bring them all the way back. So there was not a lot of food security in terms of nutritious foods beyond what they could grow right there.
Yeah, I'm thinking about your work now and how it contributes to kind of sustaining our food supply and stabilizing crops. When I think about agriculture, a lot of Americans have a particular idea of what that looks like, what a farm looks like, what agriculture looks like. And yet a lot of folks like this in Panama, these subsistence farmers or these farmers that agriculture is their livelihood, based on what you're saying, it's affected by yields every year and by if a pathogen comes in and wipes out your crop. Like that really affects your livelihood. So I guess as we're going to be thinking about ways that our climate and human activity is changing farming practices, and it's not just for mega farms that we have in the US, we're really talking about individual lives that rely on the stability of their crops. I could see how you seeing that firsthand would definitely motivate your journey a bit.
Yeah, thank you for taking me all the way back to complete the loop. I just told you this whole story about me scoring to not get back into why I got into science. So let me close that loop. So I had that experience, but I knew also that over the last, couple of hundreds of years, people have really been focused on plant breeding. And now with the advent of genetics and genomics technology, plant breeding is a highly commercialized, very competitive field, especially for food crops. So although these plants were not doing well in this environment, it's very possible to be able to create plants that can thrive in that environment and that can support the nutrition of the families that live there and that are growing them. So when I came back to the US, I decided to get a second bachelor's degree and then go straight into a PhD in genetics and genomics, focusing on plant breeding.
Wow. And this is all without a science background. You hadn't studied science formally before this?
No science background at all. It's a love of nature and of agriculture, but no formal science training.
Yeah, wow. Again, another indicator to Amina here and our other high school interns that, change is possible. You can always pivot and alter your course when a new career goal comes to mind.
Right.
So,
and you only know what you know at the time. So,
absolutely.
And you're only interested in what you know. So as soon as you learn something new, a lot of people, I think, have that feeling of, oh, well, I've invested so much in a certain point, or I had made a certain plan. I don't want to change it because I want to stay, focused on what I thought I wanted to do. But as you learn more things, it's totally fine to pivot. I think it brings a lot more richness to people's lives to be able to be curious and investigative and not just like live in a straight line, but to kind of like go in a more of a meandering scenic route.
No, absolutely. I'm sure you went into that PhD program with just a very unique perspective and experience compared to some of the other students maybe who had just been studying biology and genetics their entire lives, preparing for this. Can you tell us a little more about your PhD experience? What were you studying there?
Yeah, so I ended up coming to California for my PhD. I was accepted into UC Davis and wanted to work specifically on plant science. So based on my interest in what I had seen in Panama, I wanted to study climate adaptation in crops. And I worked specifically in chickpea, which is a really important crop worldwide. So it's very rich in protein. A lot of people grow it for subsistence farming. It's considered extremely important for human health and food security. And I ended up studying it specifically to understand climate adaptation for chickpea. So how can we still grow chickpea in a climate where it's extremely hot, where there's less water, or the water that we have is dealing with some degree of salinity, so there's added salt. So for me, it involved growing these different varieties of chickpea that were collected from around the world, and I was growing them in different fields in the Central Valley, specifically focusing on areas where it would get very, very hot, to be able to see how do these plants react under stress? How many grains are they going to produce at the end of the season? And then the plants that were the most successful, and I wanted to better understand the genetics behind this tolerance. So I ended up investigating that for a while, and then at the same time starting a breeding program for chickpea, which involves crossing the varieties that are performing the best to be able to create a variety that hopefully has the strength of both of the parents.
So you're cross-pollinating different varieties in the hopes of producing this new generation that has different, better genes. Is that right so far?
That's exactly correct, yes.
How did all of that then guide your next steps, and how'd you end up at Salk?
What I took away from that experience is that it's one thing to be able to complete some basic science. It's another thing to be able to apply it in a meaningful way. So at the time while I was working on my PhD, I was also taking MBA classes also at Davis, and it was a very different experience than going through the science. The science is very rigid. It has to be by the nature of being able to uncover something new. You have to have a strenuous application of science in these highly controlled environments. And then business school is the complete opposite of that. A lot of the time, it's very messy. You have to think about people, about motivations, about policies, about technology. So you have to be able to take everything involved to be able to essentially solve a lot of these business problems that we were dealing with in these classes. And I really liked it. I liked that it was very messy and that there wasn't a right answer. So I ended up wanting to combine the experience I had in these courses with what I had come to Davis to study in my plant background.
So I can see how then that led to your role here at Salk as the program manager of HPI. Before we go into kind of the details of the program, can you tell us a little about your role?
So I work with the now 7 different professors that make up HPI along with their labs. And I work alongside them to be able to understand and guide what our strategy looks like currently and then into the future, as well as to coordinate and execute on logistics, which is a nice way of saying fill out a lot of paperwork.
Sure. What is the Harnessing Plants Initiative and what problem is it trying to solve?
Yeah, the Harnessing Plants Initiative is focused specifically on climate change mitigation. So if we start thinking about, climate has been changing throughout the lifetime of the planet, throughout the history of people on the planet, climate change was happening, but it's happening at a very slow pace. So about like 1/3 of a degree Celsius, maybe 1/2 degree Fahrenheit, which isn't a big change over 1,000 years. But what we're seeing now that we've started really emitting carbon dioxide in significant amounts, we've seen a 1.5 degrees Celsius change in about 200 years, which is super accelerated. That change would take about 5,000 years otherwise, according to these natural fluctuations that we've been seeing. But now it's really ramped up. And right now we are at the 1.5 degrees Celsius mark that previously the United Nations set as like the maximum safe allowable amount of climate change. But we haven't really reduced our emissions in any appreciable way. If anything, they've gotten higher.
Salk can't exactly change global emissions, but we are kind of looking to impact things on the other end, on carbon sequestration. So can you tell us a little bit about what that means?
Yes. So carbon sequestration is taking carbon out of the atmosphere. So there's been a lot of talk about reducing emissions. That's part of like a big part of the climate change conversation, but also carbon dioxide removal is another part of it. And that's exactly where the Harnessing Plants Initiative is located.
So plants are naturally doing that. And again, this initiative is looking to, in similar ways to your previous projects, you're looking to develop new generations of plants that can do this even better. And I know that there's kind of two-pronged approach. Can you tell us a little bit about what those main goals of the initiative are?
Yeah, so we're focused on 2 different programs. One is our coastal plant restoration program, and then our other program is called CROPS, which is CO2 removal on a planetary scale. And that one's focused on specifically working with crops that are produced over vast amounts of land globally. So this would be considered kind of commodity crops. They're crops that are really important for humans to be able to have enough, calories to be able to survive. They include like rice, wheat, corn, sorghum, soy, and canola.
If you're enjoying this episode of Beyond Lab Walls, be sure to check out our other channels at salk.edu. There you can join our new exclusive media channel, Salk Streaming, where you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine Inside Salk and join our monthly newsletter to stay up to date on the world within these walls.
Coastal plant restoration is focused on examining these populations to be able to see if they have some of the traits that we're interested in that can contribute to these plants sequestering more carbon out of the atmosphere. And those traits involve increased root biomass, which is just the total amount of roots that are generated by a plant, increased depth of those roots, so how deep can they grow into the soil, and then are they producing specific molecules that we're interested in. These molecules are carbon rich and they're very durable. So after the plant dies and it degrades and kind of the molecules are left in the soil, these molecules are known to endure for quite a long period of time.
So by selecting for plants that have some of these traits, these larger, deeper roots or more of these carbon sequestering molecules, then those versions of the plants would be able to not only take in more carbon, but actually store it in the soil for longer. Is that right?
Exactly. And a lot of our wetlands have been degraded, especially in California. We've lost about half of our wetlands through development, but there's also an effort to be able to restore these wetlands. So by identifying populations which are native to California or to other areas where they're doing restoration work, and to be able to say these populations are specifically suited for also increased carbon dioxide removal, we can inform these restoration projects by providing these populations to them.
So you can choose those plants to be the ones that we're restoring these areas with.
Exactly, right? Exactly. We like to call it genetically informed restoration.
Oh, so cool. And then on the other end, you're looking at agricultural crops and thinking about ways to help these crops that are so important to our food supply across the world be also, again, more resilient to climate change, but also kind of do some work while they're growing, right? Help us sequester this carbon even more. So is it similar traits that you're looking for there?
That's exactly right. So we're looking for similar traits. Again, like, are they producing more roots? Are they producing roots that can grow deeper into the soil? And are they producing these very durable molecules? And I can give you kind of like a story of what that looks like, and we can go through the process a little bit.
Yeah, no, absolutely.
So we'll start with a project. Let's say it happened a few years ago, and the scientist wanted to understand what exactly causes a plant to grow deeper roots than other plants within that same species. So the scientist may first work with a model organism like Arabidopsis thaliana, which for the plant people is our equivalent of the white lab mouse. So within Arabidopsis, and there's a lot of different pools of diversity, and scientists have access to these pools, they're able to observe and phenotype them for their experiment. And they're taking notes on them. They're seeing that some of these roots are naturally growing larger than others, even though these plants are all exposed to the exact same uniform environment.
Sure.
And they're very curious about why that's happening. So they found the phenotype that they're looking for, but they don't know the gene that's linked to it yet.
Right.
So in parallel, they're going and they're sequencing the DNA of each of these populations or varieties so that now they have two different pieces of information. They have the genotypic information and the phenotypic information. And then they combine them and look to see if there are any patterns that stand out to them. So an example would be, well, look at all of these large rooted varieties. The one thing that they have in common is that there is a gene missing that is present in all of the small rooted varieties. And at that point, you get very curious about that one gene. So what does the absence of that gene mean? The scientists would then go on to do experiments specifically working on that gene to test if it is the one gene that's responsible for this difference between the two subpopulations or the two groups of like big rooted and small rooted plants. And once they have that, then it gets really exciting. So let's say that they do have this gene and it is responsible for increased root size in Arabidopsis. That's great. We've learned something for basic science, but how do we apply that in crops? So now we go through a team of bioinformaticians that have similar genetic data about all of the crop species that we work with. And they start to say, well, is this gene present in this crop? Is it present in this crop? Is it present in this crop? If it is, can we start to work with it to be able to change it? So if it is present, then we can go into the species, let's say canola, and say, okay, well, in canola, we found a gene that looks very much like the gene in Arabidopsis. Let's edit it with CRISPR. And what that means is to go in and change a few nucleotides. So an example I like to use is, let's say that the gene says cat, we can change it to bat, or we can change it to chat. So we're just changing a few nucleotides within that gene so that it's no longer operational. It speeds it up the process from traditional breeding, which can take anywhere from 5 to 10 years to a couple of years with these new genetic modification techniques. And then we grow that plant and we phenotype it to see if it does indeed have that same phenotype that we observed in Arabidopsis in the model organism. And then also to make sure that there aren't any unintended changes. So maybe that gene is responsible for some other aspect of the plant that wasn't studied previously. And if it is, that'll show up. And we'll say, okay, this gene was very interesting. Maybe we need to modify it so it's only not expressed in the roots, or maybe it's not a good candidate for us. So we've repeated this hundreds of times across many different experiments and with different crops to be able to kind of come up with this very long list of promising gene candidates that we're now working on in many different crop species. It's not trivial to build out a platform that can handle tens of thousands of plants and to be able to phenotype them for many different traits, but it's what we've been able to do over the past 5 years, and is really exciting that now we're at a place where we are making it even more high throughput, which means that we can look at more phenotypes, more plants, more species over time. So we're really accelerating the pace of the science here, which is needed because climate change is an imminent threat.
So far, we've been talking about different ways to produce new varieties through these cross-pollination techniques, breeding things out in the field. But like you just alluded to, it takes a long time. Each generation of those plants has to grow and create new generations. You're not really in control of what genes that next generation gets, like how much better it really gets in that one generation. So it might take many generations before you really get that collection of traits that you're looking for. And that sounds very different than using CRISPR, these new techniques. You already kind of know what gene you're looking for, and you can go directly into the DNA of these plants and get the genetic effect that you're looking for. So that's a major transition in the field.
It is. I think it's really revolutionized what's possible in terms of agriculture and plant science. And I think that's what we need at this point, where, again, we're racing against the clock for climate change. So being able to use these techniques and that these techniques are available to us at this time is kind of a really beautiful moment of serendipity for us. So where we are now is, let's say this researcher was doing this a couple of years ago, which they were. Now we have the canola plant that has more biomass, and that's being tested now in the greenhouse. And what that means for us is that the next stage is, once it's fully evaluated for all of the traits that we care about, the next step is to be able to assess it in a field. So it works great in a greenhouse, but how about in the actual agricultural field where the farmer will be growing it? Let's test it there too. So we work with commercial companies, nonprofits, and NGOs to be able to take the plant from the greenhouse to the field to be able to test it so that it can reach its final step with us before it finally goes on to be commercialized and used at scale, which is where it will have that real impact.
Yeah, amazing. So what are the next steps for the Harnessing Plants initiative? When will we see these plants out in the world?
Well, we're hoping that right now, with our earliest plant varieties where we've been testing these improved varieties where we've improved their ability to sequester additional carbon, we're expecting that those will be commercialized in a few years, let's say around 5 years. So for carbon sequestration or carbon removal, working with a nature-based system, once it's available, can be done at a scale that rivals anything else that's possible within carbon removal. So for instance, if you have to build a factory that's pulling carbon dioxide mechanically out of the air, every time you build a factory, you have to start from scratch. You have to build another factory and another one. But a plant, let's say you have a corn or a maize plant, you have one seed, but at the end of the season, you're going to have hundreds of seeds just from that plant. So if you continue to scale that, it scales exponentially over time, which is one of the things that we're really excited about and is one of the things that plants do exceptionally well.
Yeah. Talk about sustainable, renewable. I mean, the plants are regenerating themselves. If we can get them to be these tools for us, then that's an amazing and probably lower cost solution than creating a lot of these huge carbon sequestration factories and things.
Yeah. One of the really nice things about sequestering carbon in the soil is that when there is an increased carbon content in the soil, it makes the soil healthier too. So it's not just taking carbon dioxide out of the atmosphere and storing it in the soil that's the end of the story. Now that the carbon is present in the soil is more resilient. It can hold more water, more nutrients are available. So by building up this bank of carbon in the soil, we're creating farmlands that are more fertile, where people, where maybe if they don't have access to fertilizers or other synthetic means, that they're going to be naturally building up the soil by using these varieties, which is really exciting.
Yeah, extra bonuses.
Earlier, you were talking about like a carbon sequestering factory. That was an analogy, right? That's not a real thing.
Oh, that's a real thing.
It is a real thing?
Yeah.
Oh.
Yep.
Is it? What is that?
The original factories were built in Northern Europe, where they could use geothermal energy to be able to power the factory. It's basically like a series of fans that have a certain technology associated with them where they can actually just capture the carbon dioxide out of the air. But it's very energy intensive to do.
Yeah, it's amazing that we're getting to use plants themselves and create these versions that are just these optimized versions of what they're already doing to sequester more of this carbon and to stabilize our food supply, which with a growing population, it's clear that this is a major goal that we need to be working on right now. It's just so cool to see this go from kind of that really basic science at the bench to this product that can really be revolutionizing industries all over the world and really changing lives, like those farmers that you worked with in Panama.
It's one of the things that is definitely a life passion for me. And I'm really blessed every day to be able to work with scientists and researchers and professors who are so focused on this problem. It makes me feel like, although we are facing some really big challenges, there are also a lot of people that are working to solve those challenges at the same time.
It's such important work. We've had and we continue to have such an impact on human disease and the work we're doing there to support human health in that way. But like you're saying, one of the biggest issues of our time is how climate change is going to affect our health. And it's just amazing that we're getting to tackle that and be part of this conversation and use our basic science to have such an important impact that way. So it's so exciting to learn more about this and to see this program continue to grow. We're excited to see its growing impact in future years. So if our listeners would like to learn more about the Harnessing Plants Initiative, you can visit our website at salk.edu. And again, I want to thank so much our great co-host Amina and of course our wonderful guest Kay. It's just been so great to learn more about you and your work. And I look forward to sharing more science with all of you next time.
Yeah, it's been a pleasure to be here with you, Nicole, Amina, and to chat a little bit about my life and also about the great work that's being done here at the Salk Institute.
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.